Okay, what we're looking at is digestion past the stomach. And so during digestion in the stomach, these two sphincters are closed. And then after things in here have been pretty well digested, this sphincter, the pyloric sphincter, begins to relax and open up and let in what was in the stomach. So the official name of the liquid in the stomach is chyme. So this is very acidic, contains these broken down food pieces, and the pyloric sphincter relaxes and then slowly lets these into the very beginning of the small intestine, which is called the duodenum or duodenum. And if we look at the function of the small intestine as a whole, it has two main functions. So the first is to continue digesting food, and then after the food is digested into its smallest components, the small intestine, past the duodenum, starts to absorb these in the ileum and jejunum. So in this very first part of the small intestine, we have a bunch of secretions coming in. So from the liver, we primarily have bile containing bile salts, which we'll talk about in a second. And in between digestion, the bile is stored in the gallbladder. So it's stored in the gallbladder, and then when there is actually food in the duodenum, it's secreted out into the small intestine. And also coming in a similar place are the secretions from the pancreas, and these primarily contain a bunch of enzymes, but together we'll just call all these pancreatic juice. The main function of the bile salts is to emulsify fats. And so looking at what does emulsify mean? If you had a big ball of fat, it would be hard to digest by enzymes. They can only act at the surface. And so the stomach mixes up all the fat so that it breaks up into these small droplets. But as you know from oil and water, really quickly these droplets would come back together and then form another ball of fat. So what the bile salts do is they have a hydrophobic and a hydrophilic part. They bind to these little fat droplets and keep them separate so that other enzymes, lipases, can get in and fully break down the fat. So in addition, both the liver and the pancreas, these secretions, they contain components that break down, that chemically digest food, but they also contain components that deal with the acidic nature of chyme. And so bicarbonate is the buffer that mostly does the neutralization of the acid in chyme. And the liver also contains bicarbonate. Your book doesn't really mention it, but these both function to neutralize the acid. So the pancreatic juice has a lot of digestive enzymes. We'll get to those in the next slide. And as well as the secretions from the liver and pancreas, the intestinal walls of the duodenum are also secreting enzymes. If we look at the enzymes, you can see that a lot of enzymes are involved and coming in the small intestine. Before the small intestine, we just have these two examples of enzymes, and hopefully you can remember that in the mouth, you have salivary amylase, which breaks down carbohydrates, and in the stomach, we have pepsin, which starts to break down proteins. So the whole point here is that we want these bigger macromolecules to be broken down into their smallest component. The smallest component is what can actually, diffuse or be transported across the membrane of the intestinal cells. So carbohydrates ultimately are broken down into monosaccharides, and intermediately we want to break these down into medium-sized carbohydrates so that essentially we can have more of these enzymes come in to break them into their smallest components. So amylase first breaks them into disaccharides, and your key here is that maltose and sucrose and lactose are broken apart by these enzymes, maltase, sucrase, lactase, into monosaccharides. So then following, proteins ultimately are broken down into amino acids. Peptidases can basically break off one amino acid at a time. So before that, we have the enzymes trypsin and chymotrypsin, which will break these bigger proteins into these smaller polypeptides. So the peptidases can get in there more quickly and degrade them into amino acids. Nucleic acids are broken down by nucleases, so these break them into nucleotides. And then other enzymes, it's not important the names, break nucleotides into their components, which are bases, sugars, and phosphates. We already talked about bile salts, which stabilize fats into fat droplets, and then lipase breaks them down into the smallest components of fats are fatty acids and glycerol. So we have 3 fatty acids and 1 glycerol for every molecule of triglyceride. So lipase breaks these apart, but then before that we have these fat droplets, which are stabilized by bile salts. So I would also just make a note that bile salts are really not enzymes. They just help function in stabilizing the fat droplets. So after we've digested molecules into their smallest components, they can be absorbed in the rest of the small intestine. So if you were looking at the lumen, or the inside of the small intestine, also it's kind of good to note that by small, we basically mean thin. And Turkish does a better job of describing the ince bağırsak, because actually the intestine's quite long, but the small intestine's quite long, but very thin. In the lumen, we have a bunch of folds, and then in these folds, we have more folds, which are called villi. And so this is a zoomed in look at the villi. And then if we zoomed in even further and looked at these cells, these have on their surface microvilli. And essentially what you have is, a lot of different folds. And so all of these, the purpose is to maximize surface area. And the more surface area of cells you have, the more molecules that you can absorb over that larger surface area. And this is a concept that will come up multiple times in biology. So smallest components can pass into the cells by methods that you can probably remember from chapter 5, primarily diffusion and active transport. So diffusion includes facilitated diffusion. There are also cases of endocytosis, but not as many as diffusion and active transport. Once these are absorbed, the basically amino acids and sugars, probably can't read that, are absorbed into the blood, and so the veins take these to the liver. We'll talk about that in a second. And your fatty acids and glycerol actually recombine as fats. They're coated with these proteins that help carry them through fluid, and they go into these lymph vessels, and lymph will eventually join back with the blood. But for now, we have 2 different routes for amino acids, sugars, and fats. So the amino acids and sugars, before flowing to the rest of the body, there are, so the veins from the small intestine, all these blood vessels, go through this one main, what's called hepatic, so the hepatic portal vein are all these vessels coming up. And so before the blood goes back to the heart, it goes through the liver and is essentially filtered. So the liver, we talked about its functions in storing glucose and producing bile, but in terms of digestion, one of its other roles is to filter the blood. So taking substances which are toxic and modifying them into less toxic forms. So you can think of alcohol. These molecules are transferred into less toxic forms that can be processed by the body. In addition, your liver converts a bunch of different molecules. So there are proteins that are synthesized, blood clotting proteins, and lipoproteins, which help transport fats and cholesterol to body cells. So then, after the small intestine, uh, the fluid flows into the large intestine or colon. The very first part is called the cecum, and then we may or may not have talked about the appendix. The appendix houses a bunch of bacteria. It really isn't that necessary. It makes some vitamins that are absorbed throughout the rest of the large intestine. But if it gets infected, it can be removed. We can live perfectly fine without it. So just like the small intestine functions in absorption, the large intestine absorbs vitamins, but mostly water. So all the water that came into the digestive tract, most of it is reabsorbed, and then what's left is very little water that comes out in feces. Feces in the rectum, and then comes out in the anus. So basically, if you have diarrhea, which is very runny stool, it would mean that too little water was taken out of the large intestine. So you end up having too much water in the feces. And constipation is when you have too little water in the feces, so too much water was taken out. All right, so that's pretty much as far as we'll go. The last small note is that in animals that are eating a lot of plants, they have these very large appendix-like structures. So there's a bunch of bacteria in here that is digesting the plant matter, and that they also tend to have longer digestive tracts where they can house all this bacteria to decompose plants. All right, thanks, that's it.